Chapter 1, Molecular Foundations of Life. Molecular Foundations of Life, DNA, Genes, and Protein Synthesis. The study of molecular biology provides the foundational language of modern medicine. At the most fundamental level, life is governed by interactions among molecules, primarily nucleic acids and proteins, that store genetic information, regulate cellular behavior, and maintain the complexity of biological systems. Although medicine historically centered on gross anatomy and clinical diagnosis, contemporary medical science demands understanding of molecular mechanisms underpinning health and disease. Advances in genomics, gene expression technologies, and molecular therapeutics have transformed diagnosis, treatment, and prevention, thereby making molecular biology indispensable to future physicians. Genetic information and DNA structure. Genetic inheritance depends on deoxyribonucleic acid, a long polymer composed of nucleotide subunits that store hereditary information. Each nucleotide contains a sugar, a phosphate group, and a nitrogenous base which may be adenine, thymine, cytosine, or guanine. Complementary base pairing, with adenine pairing with thymine and guanine pairing with cytosine, creates a double helix in which two strands run in opposite directions and wrap around each other. This elegant structure stabilizes genetic information while allowing accurate replication. DNA is organized into chromosomes, compact structures housed within the nucleus, and each human somatic cell normally contains 23 pairs of chromosomes. Genes are specific segments of DNA that encode functional products, usually proteins, which determine cellular structure and physiological activity. Gene expression and protein synthesis. The expression of genetic information proceeds through transcription and translation. During transcription, RNA polymerase binds promoter sequences and synthesizes messenger RNA using DNA as a template. Post-transcriptional modifications include splicing to remove introns, capping at the 5' end, and polyadenylation at the 3' end. Messenger RNA then exits the nucleus and binds ribosomes, where translation converts nucleotide sequences into amino acid chains. Transfer RNA molecules deliver amino acids according to complementary codons, while ribosomal RNA catalyzes peptide bond formation. The resulting polypeptide undergoes folding and post-translational modifications, producing functional proteins. Although the central dogma describes a linear flow of information, regulation is multilayered. Gene expression may be modulated at chromatin remodeling, transcription initiation, RNA processing, translation efficiency, or protein degradation. These mechanisms ensure that cell-specific gene programs govern differentiation and maintain tissue identity. Genetic variation and mutation. Genetic variation arises through mutation, recombination, and chromosomal rearrangements. Mutations include point substitutions, insertions, deletions, or larger structural changes. Some mutations are silent, altering nucleotide sequences without changing amino acids, while others disrupt protein structure or function. Mutations in germ cells may be inherited and contribute to congenital disorders, whereas somatic mutations accumulate during life and often participate in carcinogenesis. The capacity for DNA repair mitigates genetic damage, but failure of repair pathways leads to genomic instability and disease. Genomics, epigenetics, and molecular medicine. Genomics analyzes entire genetic landscapes to understand complex disease susceptibility, drug responses, and population variation. Epigenetic mechanisms, including DNA methylation, histone modifications, and non-coding RNAs, alter gene expression without modifying nucleotide sequences. These processes respond to environmental inputs, diet, stress, and aging, illustrating interactions between nature and nurture. Modern diagnostic technologies incorporate polymerase chain reaction, next-generation sequencing, and molecular imaging. Clinical applications include prenatal screening, cancer mutation profiling, infectious disease detection, and organ transplant compatibility testing. Pharmacogenomics individualizes therapy based on genetic variation in drug metabolizing enzymes, improving efficacy and minimizing adverse reactions. Clinical relevance and human disease. Molecular biology illuminates the basis of inherited disorders such as cystic fibrosis, thalassemia, and sickle cell disease. It clarifies mechanisms of oncogenesis, where mutations in growth-regulating genes produce uncontrolled proliferation. Viral replication, including that of hepatitis and HIV, depends on molecular interactions susceptible to antiviral intervention. Autoimmune disease emerges from misregulated gene expression and disturbed tolerance pathways. Molecular tools also guide development of monoclonal antibodies, gene therapy, and mRNA vaccines, which recently demonstrated transformative impact in global public health. For medical learners, molecular reasoning extends beyond memorization of pathways to understanding disease at its origin. Mastery equips clinicians to interpret laboratory assays, counsel patients regarding genetic risk, and evaluate emerging therapies. As medical practice evolves, molecular biology remains a cornerstone linking basic science to clinical innovation.